Hello and welcome to my YouTube channel, AnatomyMentor. So I'm going to start now a series of lectures on the development of the cardiovascular system. Now, since this is a very long, detailed, and lengthy topic, I'm going to summarize it by dividing it into sections. And in the first part, we're going to be talking mainly about the formation of the heart tube, and I'll also discuss cardiac looping. I have some videos to make it easier for you to understand. So let's start with the first part of this series of lectures on development of cardiovascular system. So today's learning objectives will be that at the end of this discussion, you should be able to explain the formation of the cardiogenic region and consequent positioning and development of the heart tube. And also, you should be able to describe the mechanism of cardiac looping. We'll discuss some congenital anomalies. And also, we'll talk about the contribution of bulbous cordis and sinus venosus to development of the heart. Now, starting with this topic, when we talk about the cardiovascular system, you have to keep in mind that this is the first major system to function in the human embryo. And the primordial heart and vascular system, they make their appearance in the middle of the third week of gestation because the rapidly growing embryo can no longer sustain its nutritional and oxygen requirements by diffusion alone. So if you keep that in mind, you would understand how important this system is for the survival of the embryo. And there are a few basic tissue components, the embryonic tissues that contribute to formation of this heart tube, and that is splanchnic mesoderm number one. It forms the primordium of your heart. And then you've got the paraxial and lateral mesoderm, which are present near the otic plecodes. Then, of course, neural crest cells. They are going to migrate and from the region between the otic vesicles and caudal limits of the third pair of somites. So these are the embryonic tissues and also pharyngeal mesoderm. It also makes a contribution to formation of our cardiovascular system. Now, the first thing that we're going to discuss is the establishment of a region which is known as the cardiogenic field. Now, this is a region which is established in the very early stages of the human embryo by cardiac progenitor cells. These cardiac progenitor cells, which literally means the cells which are going to form the cardiovascular system, are present in the epiblast. If you recall from previous lectures of general embryology, in the second week of human development, the human being is the two-layered disc, epiblast above and hypoblast below. So the cells which are present in the epiblast, they are going to migrate through the primitive streak. Now the primitive streak was the groove on the upper surface, that is the dorsal aspect of the germ disc. And now these cells, as they will move, they will then position themselves rostral or anterior to the oropharyngeal membrane and the neural folds. So these are not unfamiliar terms if you've already gone through the lectures of general embryology. The oropharyngeal membrane, I'll just mention it again, is the part in the cranial region of the embryo where the ectoderm is lying in direct apposition with the underlying endoderm. So anterior to oropharyngeal membrane, the cardiac progenitor cells are going to settle down, and this migration occurs in a craniocaudal sequence. So here, in diagram A, you can see this is the dorsal aspect of the slipper-shaped embryo, and this upper surface over here, this is where the epiblast is present, and you can see the caudal region, there is a groove in the midline. This is known as the primitive groove, and surrounding it, the elevated edges, collectively, this is known as primitive streak. And at the anterior end, there is a depression or a pit, which is known as that is the primitive pit. And since its edges are elevated, this is known as primitive node. Now you can see that the epiblast cells are going to move through this primitive streak, and then they are going in the cranial direction. The arrowheads are showing the direction of migration of cardiac progenitor cells, and they're going anterior or rostral to this here area, which is the oropharyngeal membrane. So they're going to settle down over here. If we take a cross section through this slipper-shaped embryo, and then we are looking at it from another view, which is shown in this diagram B, we can see the epiblast above. These are the columnar cells, and below is the hypoblast. These are the flat cells. And in the epiblast, you can see through the primitive streak, here these cells are migrating. Now, these are the migrating cells which are going to form mesoderm. And few of these cells, as I mentioned, are going to migrate cranially. They're going to migrate ahead or rostral to the oropharyngeal membrane. These cardiac progenitor cells will establish a horseshoe-shaped cluster of cells, which is known as primary heart field. Now, here we can see the primary heart field. Then again, this is a dorsal view of the slipper-shaped embryo, and we can see that these patches of red are actually showing us the cardiac progenitor cells, right? So they migrated from this caudal region where the primitive streak was present, and here this is their migration as they invaginated through the primitive streak into the visceral mesoderm. They are moving cranially on both sides, right and left, and then they are going to the head region. And here, this horseshoe shape simply means a U shaped region has been established. This is known as the primary heart field, abbreviated as PHF. And this process is beginning early in the third week of intrauterine life. Now, the cardiac progenitor cells, they reside in the splanchnic layer, that is the visceral layer of the lateral plate mesoderm. If you recall from the previous lectures on embryology, the mesoderm, it subdivides into three parts: the paraxial, the intermediate, and the lateral plate mesoderm. And the lateral plate mesoderm itself is consisting of somatic and visceral layers. Well, it is the visceral or splanchnic layer, also known as the splanchnic layer, where the cardiac progenitor cells are going to settle down. Cells of the primary heart field are going to form the atria, the left ventricle, and most of the right ventricle. So these are the chambers of the heart which are formed by the primary heart field. The intraembryonic cavity, which is present over this region, will later develop into the pericardial cavity. This is the cavity surrounding the heart. So now over here, then again, we are looking at the embryo in the early stages, early weeks of development, the dorsal view, where this horseshoe-shaped or U-shaped region is the primary heart field, which is present cranial to oropharyngeal membrane, cranial to these neural folds. And in the caudal region, we have the primitive node and primitive streak. So then again, if we take a cross section through this embryo, and then we can see all these three layers. If it's the third week of development, you can see the ectoderm above, the endoderm below, and in between is the mesoderm. And as I told you, the mesoderm subdivides into the most midline one, which is paraxial mesoderm. Intermediate mesoderm is lateral to it, and then eventually is the lateral plate mesoderm, which splits into these two layers. The layer which is adjacent to ectoderm is the somatic or parietal layer of lateral plate mesoderm, and the layer which is adjacent to endoderm, this here is the visceral or splanchnic layer of the lateral plate mesoderm. And this is the layer where these cardiac progenitor cells have reached and where they are forming the primary heart field. So this is the primary heart field. In this diagram, you can see it is U-shaped as you are looking at it from the superior view, that is the dorsal surface of the embryo. And if you have taken a section, then this is a cross section. So here you can see the primary heart field on both the left and the right side. And then in C, you can see the other section, which is known as the sagittal section. This would be in case of if we are slicing this embryo in the midline, and then we are looking at the sagittal section. So here in the sagittal section, then again, you can see that this is the ectoderm, and this is the endoderm, the intervening mesoderm, and cranial to this ectoderm, you can see over here is the oropharyngeal membrane, where I told you the oropharyngeal membrane is the place where the ectoderm is directly attached to underlying endoderm without intervening mesoderm, and cranial to this, that is rostral to this oropharyngeal membrane, you've got the primary heart field. And just above it, the part of intraembryonic cavity which is lying above it is forming the pericardial cavity. So in the sagittal section, we can see that this primary heart field, it has been shown from this area over here specifically that it is lying anterior to your oropharyngeal membrane. Now, what about the rest of the heart? That is the part of the right ventricle, outflow tract, atria, and sinus venosus. They are derived from another region which is known as the secondary heart field, abbreviated as SHF. It consists of cells of splanchnic mesoderm, which are ventral to pharynx, and this is located medial to the primary heart field. So this here is the region of the secondary heart field, and you can see that it is ventral to the ventral to the pharynx. And over here, it is contributing, the cells from secondary heart field are contributing to the outflow tract of the developing heart. Cells of the primary heart field, they are induced by underlying pharyngeal endoderm to form cardiac myoblasts and blood islands that will eventually form blood cells and vessels by the process which is known as vasculogenesis. These islands will then unite and they will form the horseshoe-shaped endothelial lined tube surrounded by myoblasts. So myoblasts are the cells which are going to form the cardiac muscle. This region is known as cardiogenic region. So if we take a section from this slipper-shaped embryo, and this section will show us how there is a sequential development of the mesenchymal cells into the blood cells and into the endothelial cells. So initially, these mesenchymal cells, they are you can see that they are distributed randomly, but then they become organized and then they are becoming differentiated and they are rearranging themselves, and those cells which are in the center are going to form the blood cells, and those cells at the periphery, they are going to attach and they're going to form the endothelial cells, and this is how the blood vessels are going to be formed. This process of formation of blood vessels from these blood islands is known as vasculogenesis. Other blood islands appear bilaterally, parallel and close to the midline of the embryo, and these islands form a pair of longitudinal vessels known as the dorsal aortae. Now let's talk about the formation and position of the heart tube. So remember, during cardiovascular development, the heart tube is a structure which is formed before the heart. So first you get the heart tube, then the heart tube is going to fold upon itself, and then we will get eventually the adult appearance and formation of the heart. So cephalic folding. This, during the fourth week of development, there is folding of the embryo in the cranial and caudal regions. This folding, which occurs in the sagittal plane, that is going to be responsible for the formation of the head and tail folds. So the folding in the head region, the folding that is known as cephalic folding, pulls the oropharyngeal membrane forwards, while the developing heart and pericardial cavity move ventrally in the region of the thorax. So let's go back to this diagram that I showed you. This is a smaller section from the larger sagittal section of the disc-shaped trilaminar germ disc. So this here is the ectoderm, which has formed the primordium of the nervous system. Below that is the mesoderm and then the endoderm, and the mesoderm is forming the notochord. So this section is a sagittal section and the head region of the developing embryo in early stages of development. As I showed you before, anterior to the developing embryo, this here is the oropharyngeal membrane, and then anterior to the oropharyngeal membrane, we had the primary heart field, which, as I told you, because of vasculogenesis, it has now formed the heart tube. And just above it, the part of intraembryonic cavity, which is present, has formed the pericardial cavity. Now, what we are trying to explain over here is that the process of cephalic folding, which occurs in the head region of the developing embryo in fourth week of development, because of that, this area over here is going to move downwards, or ventrally, you could say, in this direction of the arrowhead. So if you look at the next diagram in the sequence of events, which is diagram B, this portion of developing heart surrounded by pericardial cavity has now moved below or ventral, and eventually it has come to lie caudal to the oropharyngeal membrane. So this is because of the formation of the head fold. This is a repositioning of the developing heart tube. Body wall folding in fourth week of development also occurs in the transverse plane, that is known as the lateral folding. This results in a fusion of the caudal regions of the paired cardiac primordia, except at the caudalmost ends. And this eventually leads to formation of the heart tube. So the folding in the lateral plane, in the transverse plane, this is going to cause the paired cardiac primordia to fold. And this here is a small video which is going to show us how this process occurs.
The flat embryo folds along its long axis, thus giving the embryo its tubular shape. The lateral endocardial tubes come together, as a result of the folding, ventral to the embryo and fuse to form a single heart tube.
So you can see the paired endothelial tubes, they have fused and they have formed this single heart tube. And these are the dorsal aortae, the paired dorsal aortae. Now, these dilatations are representing the parts of the heart tube in the middle. Okay, so remember the folding in the transverse plane, that is the lateral body wall folding, is going to cause the fusion of these paired cardiac primordia. This was the horseshoe shaped or the U shaped cardiogenic region. And because of the folding, as you can see, the direction of these arrows are showing you the direction of the lateral body wall folding will cause these paired cardiac primordia to come together over here. These are the paired endocardial tubes. As they come together, they will form a single heart tube in the midline of the embryo. And these are the cross sections which have been taken through the embryo and are showing us the fusion of the paired cardiac primordia. So these are the diagrams. You must be familiar with them if you've attended the lectures on general embryology. These are showing us the cross sections which have been taken, and they are taken through this trilaminar germ disc. And above you can see the amniotic cavity, below is the yolk sac cavity. This portion here represents the trilaminar germ disc in cross section. Above in blue is the ectoderm, then you've got the mesoderm, and below in yellow you've got the endoderm, and here in the midline is the notochord. So if we look at the lateral plate mesoderm, as I told you, it has a somatic and visceral layer, and within this visceral layer, also known as splanchnic mesoderm layer, are the angiogenic cell clusters. This was the region where the primary heart field is established. So over here you can see now, as lateral body wall folding is going to take place, you can see over here these are the folds which are moving ventrally. This brings these paired cardiac primordia closer to each other towards the midline, and so they fuse, and if you look at diagram C, they form this single heart tube. And the body wall folding in the lateral plane has caused this fusion or this closure of the ventral body wall. And here now you've got the endocardial, we can simply call it the heart tube, and it is surrounded by the cardiac myoblasts, which are going to form the myocardium. And initially there's also a gelatinous material known as cardiac jelly, which disappears later on. Also, remember, initially this heart tube is attached to the body wall by this here fold of tissue, which is known as the dorsal mesocardium. This is also going to disintegrate in later stages. Also, this heart tube is suspended in this cavity. This part of the intracavity which is surrounding it is known as the pericardial cavity. Now let's talk a little about the layers of the heart which are going to be formed within the heart tube. The innermost layer, the endocardium, is derived from the inner endothelial lining of the heart. Then comes myocardium, which is a thick muscular layer derived from cardiac myoblasts. And this myocardium secretes a thick layer of extracellular matrix called cardiac jelly. It is rich in hyaluronic acid and it separates it from the endothelium. The epicardium or visceral pericardium is derived from mesothelial cells, and these mesothelial cells, they arise from the external surface of sinus venosus and spread over the myocardium. Now, these series of diagrams have been taken from Creethel Moore, and they're showing us the ventral view of the developing heart and pericardial region. So if you look at the development of the heart from the ventral view, you can see here is the pericardial sac and the pericardial cavity, in which we have these two paired endocardial tubes, and then they fused in the midline to form this single heart tube. As a result of lateral body wall folding, they fuse to form a single heart tube. And now different parts of this heart tube will become dilated, and they will form our heart eventually. And this structure right now is called the heart tube. It is going to fold upon itself and it will form the heart. And also you can see the layers. If you want to look at the layers of the heart, you can see the endocardium, and you can see the you can see the endocardium, and you can also see the myocardium. And then externally, we're also going to talk about the layers of the heart. This is a process which is, as I told you, I'll talk about it later in detail, the folding of the heart tube. This heart tube, you can see, is going to bend. It is going to bend in such a way that this lower or caudal part of the heart tube, which is known as the sinus venosus, and just above it is the primordial atrium. This part is going to move upwards or cranially, while this part, which was the cranial part, original cranial part of the heart tube, that is the region which is known as truncus arteriosus, it is going to move downwards and caudally. So the heart tube is going to bend. Now, this is a schematic diagram which is showing us the heart tube in the early stages of development. And this is a very important one. I'd like to especially tell the students that they should be able to draw and label this diagram because it's going to show you the main parts of the heart tube. And this could be useful in your OSPE exams and can also be asked during your Viva exams. So this one is something that you should know from the top of your head. Now, these are the parts of the heart tube in a craniocaudal sequence. Well, over here we've got the cranial-most part, which is the truncus arteriosus, and then below that is the bulbus cordis, and then caudal to that is the largest dilated portion of the heart tube, which is the primitive or primordial ventricle, and caudal to that is the next dilatation, which is the primitive or primordial atrium. And caudal to that is the sinus venosus, which is receiving the two horns, right and left horns, and each horn itself is formed by the entrance of three veins, namely the common cardinal vein, the umbilical vein, and the vitelline vein. So you can see over here that at the uppermost part of this heart tube is the aortic sac, from where are exiting the arteries which are going to enter the pharyngeal arches, so they're called the aortic arches. Now, the layer which is surrounding this heart tube, lining it, is the visceral layer of serous pericardium, also known as the epicardium, and it is continuous with the parietal layer of the serous pericardium. And between these two layers is the area which is that space is known as pericardial cavity, which has a small amount of pericardial fluid. And on the outer aspect is the tougher fibrous pericardium. So this is the diagram which is showing us the parts of the heart tube in a craniocaudal sequence in early stages of intrauterine life. Now, if we were to make a flow chart, then we could see that all of these parts, they are actually channeling blood in specific direction. Right now, you've got to remember that initially this heart tube is suspended in pericardial cavity by a fold of tissue known as dorsal mesocardium. This will eventually disappear and it will be replaced by a passageway or space known as transverse pericardial sinus behind the heart. And the heart itself is suspended by the blood vessels at its cranial and caudal ends. So this here is a cross section of the embryo, which is showing us the dorsal mesocardium, which is suspending the heart tube in the pericardial cavity. And this heart tube is surrounded by the myocardium and the cardiac jelly. So this dorsal mesocardium will eventually break down, and this is a sagittal view showing us the bulging heart tube suspended within the pericardial cavity. Now let's look at the blood vessels, the initial framework of the blood flow in the early stages of embryonic development. This region is the heart tube. Now let's summarize again in a craniocaudal sequence the parts of heart tube, which are the truncus arteriosus, then you've got the bulbus cordis and the primordial ventricle, then they've got the primordial atrium and sinus venosus. Sinus venosus is receiving these three sets of veins. You can see the common cardinal vein is formed by union of anterior cardinal and posterior cardinal vein. It is bringing blood from the body of the embryo. And then you've got this here umbilical vein, which is bringing the oxygenated blood from the placenta. And then the vitelline vein, which is bringing the blood to the embryo from the yolk sac. As this blood enters the caudal part of the heart tube, it will travel through it, and from the cranial end you can see the aortic sac. Here emerge the aortic arches, which are numbered according to the pharyngeal arch in which they are located, and then the blood enters this here dorsal aorta, and also you can see the umbilical arteries. They are going to carry the deoxygenated blood away from the embryo to the maternal placenta. So the blood that enters sinus venosus, it is entering from the embryo, placenta, and yolk sac, and in the heart tube from the sinus venosus, it is moving through the primordial atrium and AV canal to primordial ventricle, and then bulbus cordis, truncus, aortic sac, eventually into the aortic arches, and then again to the embryo, the yolk sac, and the placenta. Now, the heartbeat begins at day 22 to 23. So it's in the fourth week of development when blood flow starts, and it can be visualized by Doppler ultrasound. So here is the Doppler ultrasonograph, and you can see the area in red over here is representing the blood which has been detected and in the early stage of the embryo in the fourth week of development. And the circulation through primordial heart, remember initially the contractions are of myogenic origin, and the muscle layers of atrium and ventricle, they are continuous, and peristaltic wave-like contractions begin in the sinus venosus. And at first, this circulation is ebb and flow type, which means, like you would say, the ocean waves which touch the shore as they come and go. But then by the end of the fourth week, coordinated contractions of the heart, they result in a unidirectional flow. Skipping these exercises, now let's come to the important topic of cardiac looping. Now, I told you before that the heart is going to bend upon itself. This bending takes place in a right-sided direction, so it's known as the dextral bending, and it occurs in the fourth week of development, and it results in a U-shaped or D-shaped loop with the apex of the heart pointing towards the left. So in the adult heart, the heart is slightly placed towards the left of the chest cavity, and the apex will be pointing towards the left, and it is lying in the fifth intercostal space. The heart tube begins to fold or bend upon itself on day 23 of gestation. In this case, the superior part, that is the arterial or cephalic portion, bends ventrally, caudally, and to the right, whereas the caudal, that is the inferior or venous portion, shifts dorsocranially and to the left. This creates the cardiac loop, which is completed on day 28. Now, here are the series of diagrams. They've been taken from Langman's Medical Embryology, and they're showing us the bending of the heart tube. And here I've drawn a compass just so you get an orientation of the direction that this is superior and then inferior. And on this side, you've got the left, and here is the right. So in the early stage of the heart tube is when it is not bent. Let's look what happens in the upper portion. This upper portion I told you in green over here is the bulbus cordis, and in black you've got truncus arteriosus. Now, as you can see, this portion is moving downwards. It is moving downwards over here. It is also moving ventrally, and it is moving to the right. Whereas the lower portion, which is the part of the sinus venosus and the atria, this portion you can see is moving upwards dorsally and to the left. Also, you can see because of this bending, the atrium, you can see, which was initially outside the pericardial cavity, is now present within the pericardial cavity, and its boundaries are represented by this broken line. And as a result of this cardiac looping, also remember the atria, as they're moving upwards, they come to lie in a more dorsal position, whereas this ventricle, you can see, and the conus cordis, they come to lie anteriorly with respect to the atria. So the results of looping is that, as I said, the heart invaginates into pericardial cavity. The atrium and sinus venosus come to lie dorsal to the truncus arteriosus, bulbus, and ventricle. Why does looping occur? This phenomenon occurs because of disproportionate growth in the different parts of the heart tube. Because the bulbus cordis and ventricle grow faster than other regions, the heart tube will bend on itself, forming a U-shaped loop. There are certain changes which are going to occur also now in the bulbus cordis. Its proximal dilated portion will be trabeculated and form the primitive right ventricle. The mid portion of bulbus, which is known as conus cordis, will form the outflow tracts of both ventricles, whereas the distal part, which is truncus arteriosus, it will form the roots and proximal portions of aorta and pulmonary trunks. Also, the trabeculated primitive ventricle forms the primitive left ventricle. The AV atrioventricular junction remains narrow, forming the atrioventricular canal, and the bulboventricular sulcus, which is present between the bulbus cordis and the primordial ventricle, it remains narrow, forming the primary interventricular foramen. Folding and rotation of the heart, we talked about this before. The conotruncal portion of the heart shifts to the right side of pericardial cavity to a more medial position, leading to formation because of the formation of two transverse dilatations of atrium. As I showed you, these two transverse dilatations in red out of the right and left atria, they have come to lie dorsal to this part, which is in blue, the ventricle, and the conus cordis in green. Now we're going to understand these events by looking at a video clip which is showing us the formation of the heart tube, and also it is showing us the bending of the heart tube. So this is the heart tube, the arterial end, this is the caudal or venous end, and these are the parts of the heart tube. And you can see over here that now it has bent, so the caudal part has moved upwards and cranial part has moved downwards. Also, the caudal part, which you can see in purple, which is the sinus venosus and primordial atria, as it moves upwards, it also moves dorsally. This can be better understood when we are looking at this process also from the side view or the profile. We can see this portion in purple, which are the atria, the primitive atria. They are moving upwards and they're coming dorsal, whereas this portion in green, that is the ventricles, they are now anterior. Then again, the cardiac looping, the heart tube is now a D-shaped or an S-shaped loop, which has bent upon itself, and this is responsible for the final positioning of the chambers of the heart. Now, what happens if the heart tube doesn't loop to the right, but instead it loops to the left? Well, this will result in the heart being misplaced. Instead of being on the left, it is going to be towards the right. The apex, you can see, will be pointing towards the right. This condition is known as dextrocardia. So this is a congenital variation, an anomaly, but mostly it will be asymptomatic, and a very small percentage of the population have dextrocardia. Now, what are the changes in sinus venosus? Sinus venosus, the adult functional remnants of sinus venosus, they are derived from the horns of the sinus venosus. Each horn, I told you, is receiving blood from three important veins: common, cardinal, vitelline, and umbilical. Now, what happens is that due to the left to right shunting of blood, the left sinus horn loses much of its importance, and all that it eventually forms are the oblique vein of left atrium and the coronary sinus, whereas the right sinus horn enlarges greatly and it forms the smooth wall part of right atrium, also known as sinus venerum. The remainder of the right atrium and auricle, they are derived from the primitive atrium, that is the rough trabeculated part. So the changes in sinus venosus, you can see over here in the series of diagrams, the regions in blue are representing the horns of the sinus venosus. And as I told you, the left sinus horn is largely lost, and it only forms the oblique vein of left atrium and the coronary sinus. Whereas the right sinus horn, it gets incorporated into the right atrium, forming its smooth wall part known as the sinus venerum. So the sinoatrial orifice is flanked by right and left venous valves. The left venous valve fuses with the developing atrial septum, and the right venous valve gives rise to valve of inferior vena cava and the valve of coronary sinus. Now, what about the development of the left atrium? Most of it is formed by absorption of primordial pulmonary vein and its branches. And the primitive left atrium becomes the left auricle, a tubular appendage of the left atrium. So here you can see the series of diagrams where the pulmonary veins have become absorbed within the left atrium, forming its smooth wall portion, whereas the original primordial atrium has formed the left auricle. Now we're going to summarize the adult derivatives of the embryonic structures regarding the development of the heart. And this is a very crisp and specific summary. So let's start with the first one, and we're going in a craniocaudal sequence in the heart tube. So the proximal 1/3 of the bulbous cordis, this is going to form, the trabeculated part of the right ventricle, whereas conus cordis will form smooth outflow tracts of both the ventricles. If you look at the truncus arteriosus, which is the most superior aspect of bulbous cordis. This will form the roots and proximal portions of the aorta and pulmonary trunk. Primordial ventricle gives rise to trabeculated part of the left ventricle, and primordial atrium forms trabeculated parts of both atria, right and left auricular appendages. Sinus venosus, as I told you, it forms the smooth part of right atrium, oblique vein of left atrium, and coronary sinus. And the pulmonary veins are going to form smooth part of left atrium. So you can draw the stages of cardiac looping, and you can also watch the video, the link I've shared for the process of cardiac looping, and that would be useful. This ends with today's discussion on the formation of the cardiogenic heart field, the primary heart region, and also the formation of the heart tube, the looping of the heart tube, and we also discussed the adult derivatives of the different parts of the heart tube. And this was the first part in the series of lectures on the topic, which is the formation of the cardiovascular system. If you found today's discussion helpful, then don't forget to like and subscribe to my YouTube channel, Anatomy Mentor. And you can simply do that by just pressing on the picture that is going to appear at the end of this video. And don't forget to hit that bell icon for regular notifications and updates, because I'm soon going to be coming back with the rest of the topics. And then we're going to discuss the formation of the blood vessels. We'll be talking about the aortic arches and also some congenital heart birth defects. So take care and stay blessed, and best of luck for your exams.